In this lecture I will compare class 1 and class 2 MHC molecule, and these are the all points. The first is components. The class 1 MHC molecule is composed of alpha chain and beta 2 microglobulin, these two polypeptide. But in case of class 2 MHC molecule, it is composed of alpha chain and beta chain. So let me draw class 1 MHC molecule. Class 1 MHC molecule looks like this. So this is the beta 2 microglobulin, this is the alpha chain, and the alpha chain contains three domains: alpha 1, alpha 2 and the alpha 3. And if I draw the class 2 MHC molecule, it looks like this. If this is the alpha chain, then this is the alpha 1 domain and this is the alpha 2 domain, and if this is the beta chain, then this is the beta 1 and beta 2 domain. Now in this region, the alpha 1 and alpha 2 make a peptide binding cleft or groove where the peptide bind. And in case of class 2 MHC molecule, the alpha 1 domain and beta 1 domain actually represent the peptide binding cleft. Now the polymorphism, as I have already mentioned, MHC molecules are polymorphic in nature, but whole the structure is not polymorphic. The antigen presenting site like alpha 1 alpha 2 domain and the alpha 1 beta 1 domain are actually polymorphic. So in case of MHC 1, the alpha 1 and the alpha 2 domain show polymorphism, and in case of MHC 2 molecule, alpha 1 and the beta 1 domain, this class 1 MHC molecule is expressed by all nucleated cells. But class 2 MHC molecule is very restricted, like professional antigen presenting cells, dendritic cell, macrophage and the B cells are actually express MHC 2 molecules. Another cell called thymic epithelial cells and endothelial cells can express class 2 MHC molecule. Now the responsive T cells. Which T cell can bind with the MHC 1 molecule? It is called cytotoxic T cells, also known as CD8 positive cells, because cytotoxic T cell express a co-receptor called CD8. So responsive T cells is TC cells. TC cells or cytotoxic T cells can bind MHC 1 molecule. But in case of class 2 MHC molecule, T helper cell, also called CD4 positive cell, actually can bind with the class 2 MHC molecule. The binding site of the T cell co-receptor. In case of TC cells, the alpha 3 domain of the MHC 1 molecule. CD8 co-receptor actually bind with the alpha 3 domain of the MHC 1 molecule. But in case of MHC 2 molecule, the alpha 2 and the beta 2 make a pocket and CD4 actually bind in that pocket. The size of peptide. The range is MHC 1, in case of MHC 1 it is 8 to 10 amino acid, very short, and mostly it has been seen that the peptide that bind with the MHC 1 molecule are mostly 9 amino acid. The range 8 to 10, but most peptide that bind with the MHC 1 is 9 amino acid. But in case of class 2 MHC molecule, it is 13 amino acid to 18 amino acid, larger than the MHC 1 molecule, and it could be larger also, larger than the 18 amino acid. The next is site of antigen processing, where the antigen are processed. In case of class 1 MHC molecule is cytoplasm, and I want to mention here a molecule that is responsible for the processing of the antigen. In case of MHC molecule is called immunoproteasome, and in case of class 2 MHC molecule, the site of antigen processing is late endosome, and the responsible molecule that process the antigen are endosomal and lysosomal protease. So enzyme are responsible for the processing of actually antigen. One example of this protease is cysteine protease, very important example of this cathepsin. Now the site of antigen loading, where the antigen are loaded onto the MHC 1 and MHC 2 molecule. In case of MHC 1, the site is rough endoplasmic reticulum, but in case of MHC 2 molecule, the site of antigen loading is also the late endosome. The next point is nature of antigen. Which type of antigen are presented by class 1 MHC molecule and class 2 MHC molecule? In case of class 1 MHC molecule, vital protein, self protein or self antigen, you can write it vital antigen. Actually these are protein in nature because I have mentioned that MHC molecule can only represent peptide antigen. So if I write vital antigen, it means actually peptide, viral peptide, cell peptide or cell protein or self antigen. And one important another antigen is called neoantigen. What is neoantigen? I have already mentioned that neoantigen means when a cell transforms into a tumor, the tumor cells start to produce some new type of antigen. These are called neoantigens. So it is in tumor condition. But in case of class 2 MHC molecules, the antigen come from the phagocytosis, then endocytosis and autophagy. So these three are the route from the antigen are entered into the cell and presented by the MHC II molecule. And this all pathogen actually enter a vesicles. Like if it is phagocytosis, then the pathogen or the antigen enter into the phagosome. So this is a vesicle. If it is occurring through or the pathogen is entering into the cell through endocytosis, it is actually entered into a vesicle called endocytic vesicle. So these are the vesicle. And in case of autophagy, it is present in the autophagosome. By phagocytosis, bacteria, protozoa can be engulfed. By endocytosis, actually receptor mediated endocytosis is performed by B cells, and this phagocytosis is performed by dendritic cell and macrophage. So endocytosis, by the endocytosis, B cell can engulf bacteria or soluble antigen. I want to mention this bacteria, protozoa, these all pathogen or antigen do not come directly into the cytoplasm. They remain in a vesicle. So this is the difference. In case of class 1 MHC molecule, the antigen are present directly into the cytoplasm and processed by immunoproteasome and loaded into the RER. But in case of class 2 MHC molecule, the pathogen or the antigen is actually present in vesicles like phagosome, endocytic vesicles or autophagosome. Then it actually the antigen are processed into the late endosome and also it is loaded onto the late endosome. Antigen processing and presentation by the class 1 MHC molecule is called endogenous pathway. But in case of class 2 MHC molecule, it is called exogenous pathway. Why it is called endogenous and it is exogenous? Because in case of class 1 MHC molecule, the antigen or protein antigen is present in the inside of the cell, especially cytoplasm. Exogenous means here the antigen or pathogen or the peptide is actually present in a vesicle, exogenous structure.